is Aaron Michael, matrix number 176349. So today I'll be talking about enzymes. Enzymes are actually proteins found in our own bodies as well as other living organisms. So the function of enzyme is to speed up biochemical reactions. Now the common misconception about enzyme is that without enzymes, people think that chemical reactions will not occur. The fact is that enzymes only speed up chemical reactions. They are not 100%, how to say, it's not really involved in the reaction. They just speed up the reaction. Now, enzymes speed up reactions by lowering the activation energy of the reaction. They lower activation energy by weakening the bonds between the substrate. Excuse me. So I'm here to tell you a few things about enzymes. The first thing is that enzymes are very specific. They only fit to certain substrates, and there are two models to explain this specificity of enzyme. The first one is the lock and key model. The second one is the induced fit. The lock and key model explains that the active site of an enzyme is the exact shape of the substrate. The induced fit model states that the enzyme will change shape whenever it binds to the substrate. The induced fit model is more preferable after research has been done. The next thing I want to tell you about enzymes is that enzymes are very sensitive to temperature and pH. The optimal temperature for enzymatic activity is about 37 degrees. That is about our body temperature. At lower temperature, enzymes tend to be inactive and not be as effective at the temperature of 37. pH also affects the enzymatic activity because pH alters the the charge on the active site. So whenever the charge of the active site is different, the substrate can no longer bind to the enzyme. So that's it. Thank you.
Hi, my name is Tan Zermin and my metric number is 180353. Last week we did experiment on enzyme kinetics. Before we discuss about enzyme kinetics, we first need to know about enzymes. What are enzymes? Enzymes are biocatalysts, which are also protein molecules, which have binding sites that allow substrates to bind, and then they will speed up the reaction. So what type of molecule is an enzyme? As stated before, an enzyme is a protein molecule in its tertiary structure, and an enzyme has a substituent group which will act as a binding site for a substrate to bind to. This binding site, after binding to a substrate, will form an enzyme substrate complex together with the enzyme. And how does this enzyme speed up the reaction? After forming an enzyme substrate complex, the enzyme will lower the activation energy for the reaction, and thus allowing faster reaction for the substrate to turn into a product. So in this enzyme kinetics experiment, we use an enzyme called cello-biase enzyme. And this cello-biase enzyme is also known as cellulolase, which breaks down cellulose. An example of industrial application of this enzyme is it is used in factories to break down the cellulose of the plant to sugar, which is then made into ethanol for fuel. biofuel. Okay, so what are the natural products for this enzyme? As we know, once a substrate is broken down, we get enzymes. So the substrate for this cellulose is p-nitrophenol glucopyranoside. Okay, so after breaking down this p-nitrophenol glucopyranoside, we will get two products, glucose and p-nitrophenol. Let me repeat it, glucose and p-nitrophenol. So how do we determine the amount of product that is produced at each time period? We will use this to through this experiment, enzyme kinetics, we will use cuvettes to do this experiment and determine the amount of product that is produced at each time period. Thank you.
Okay, my name is Nur Kuain binti Hamka and my metric number is 180308. Last week, we have done this experiment that include enzyme, whereby we study about their kinetics. So at first, I'm going to explain to you what is actually an enzyme. Enzyme is a specialized protein that is very specific that can speed up the chemical reaction by lowering their activation energy. Enzyme is very specific, whereby they can only have two types of bind, which is lock and key and induced fit. Lock and key is whereby the enzyme is the lock and the substrate is the key. Means that the substrate must really accommodate the shape of the active site of the enzyme. Example, this one is the enzyme, so the substrate is this, so it's bind, same shape. For the induced fit model, the induced fit model is whereby the enzyme is actually have to fit the substrate. For example, if this is the enzyme and this is the substrate, it will actually make their shape like this, and I can actually bind with the substrate. In a chemical reaction, the enzyme can work properly in average pH and temperature, whereby a very high temperature, example more than 37 degrees Celsius, which is our body temperature, can cause the enzyme to denature. So and also the pH too acidic of pH will cause the enzyme active site to change shape, so substrate cannot bind. Okay. In our practical, we use the cellulobiase as the enzyme, which is very important in the industrial use, whereby cellulobiase is the enzyme that will the substrate is the cellulobiose, so enzyme will bind to cellulobiose. So it will produce glucose. For the glucose is for the ethanol fuel. Glucose is used for creation of ethanol for fuel. I think that's all. So thank you.
Hello and Assalamualaikum warahmatullahi wabarakatuh. My name is Siti Fatimah Zahra binti Abdul Rahim and my metric number is 180306. Okay, last week we have done an experiment regarding the kinetic enzyme. Okay, today I'm going to explain what is actually an enzyme. Okay, enzyme is actually the specialized protein which act as a catalyst to speed up certain chemical reaction. Okay, why is structure of the enzyme is very important, and why it is because the enzyme determine which the enzyme is highly specific, which it determine what product is going to produce. For example, the enzyme have this shape and the substrate have this shape, and it need to feed together so that the product can be produced. Okay, they have two types of feed, which one is a lock and key, and second is the induced feed. The lock and key taught how the enzyme is actually need to be very fit to the substrate. However, the induced feed taught that the enzyme can actually change its shape to fit with the substrate to produce a product. Okay, enzyme is affected by the temperature and pH. It needs to be in the average pH and temperature, which is our body temperature, 27 degrees Celsius, and a neutral enzyme, a neutral pH value. Okay, if there is a too high of temperature or too low of temperature, the enzyme will be denatured and it's not going to be used properly. Okay, there is three types of inhibitors. First is the competitive, second is non-competitive, and third one is uncompetitive. For competitive inhibitors, the inhibitors itself will bind to the enzyme, so it will prevent the substrate to bind to the enzyme. For example, there's only this is the enzyme. It only has one active site, which will bind only to one compound. So when the inhibitors feed the enzyme, so the substrate can no longer feed the enzyme, and therefore they inhibit the production of the product. Okay. Second time is non-competitive. What is non-competitive inhibitors? It is when the enzyme can actually feed both inhibitors and the substrate, but when the inhibitors bind to the enzyme and together with the substrate, so the inhibitors will actually change the enzyme properties so that it cannot react with the substrate and no product will be produced. And last one is the uncompetitive inhibitors. Uncompetitive inhibitors is whereby the enzyme will fit together with the substrate, and the inhibitors will bind to the enzyme substrate complex and inhibit the production of the product. And I think that's all about the enzyme, and thank you.
And good morning. My name is Amira binti Muhammad Halimi. My metric number is 180297. So, our last experiment is about determine the reaction rate improvement of the enzyme. Okay, here I want to talk about enzyme. What is enzyme? Enzyme is a specific protein. It acts as catalyst to speed up the reaction. Without enzyme, the reaction will slower. In the term of chemical, enzyme lower the activation of the energy. Okay. Each enzyme is very specific. According to lock and key hypothesis, enzyme is the lock and substrate is act as the key. Only specific enzyme, only specific substrate can bind to the active site of the enzyme. So it can produce, when it binds to the active site, it can produce the product. Okay. If the temperature of the enzyme is very high, the active site of the enzyme will denature. So the reaction cannot occur. Therefore, only reaction of the enzyme only occur at suitable or ideal temperature and pH. Enzyme don't change at the end of the reaction. That's on the experiment. Last week we used spectrometer. Spectrometer is measure the amount of the light that is absorbed by the sample. If the darker of the yellow of the sample, the more light is absorbed. Therefore, more concentration of the sample. Okay, the absorbance value of the reaction sample can be measured, and the standard curve can be used to convert the absorbance value to the concentration value. That's all from me. Thank you.
Good morning, my name is Yim Yanne. Matrix number is 180304. Okay, so 2 weeks ago we did our lab experiment about enzyme kinetics. So for the questions that we needed to answer is what type of molecule is an enzyme? So to answer this, an enzyme is actually a protein compound. It's made from amino acids. It has a quaternary structure, so meaning that it folds into its specific shape. So why is the shape important is that enzymes actually are very specific compounds. So each specific enzyme has its specific substrate to make into a specific product. So the shape actually, the enzyme actually has an active site on it. So the active site has a unique shape for that particular substrate. So an enzyme actually is a catalyst, an organic catalyst. So it speeds up chemical reactions that occurs in organic life forms. So it does this by combining with an enzyme with a substrate, and then lowering its transition state and uses less energy and less time. So speeds up the reaction. So next, for this experiment, the enzyme used is cellulobase. So cellulobase in the natural world is used to break down glucose. So for digestion. So the product is actually p-nitrophenol. So okay. So there's actually two hypotheses as to how an enzyme works. That is first the lock and key hypothesis, and the other is the induced feed hypothesis. So the lock and key, as I have explained earlier, is that specific substrate to specific enzyme. For induced feed, the enzyme's active site actually changes its shape to accommodate the binding to the substrate. So next, again in this experiment, we have to calculate the amount of p-nitrophenol, which is the product, by using spectrophotometry. So this is actually to study the effect of enzymes over time. So yeah, spectrophotometry is actually the measure of light absorbed by the sample. Thank you very much.